Hello everyone and welcome to another video tutorial from Attenuated. So today's the topic is vaccination and immunization. So today we will discuss on their types and how does they work and all other concept on vaccination and immunization. So let's move on. So immunization and vaccination, what are they? Let's see now. So immunization, deliberate provocation of an adaptive immune response by introducing antigen into the body. So basically, immunization means the process of increasing the immune response against antigen entered into the body. So this was the basic concept here. Now vaccination, deliberate induction of an adaptive immune response to a pathogen by injecting a vaccine, a killed or attenuated form of the pathogen. So in this process what happens is unknowingly the pathogen is injected into the body, which might be killed or in living form, and against to that the immune system builds a antibody. So this is the process of vaccination here. Clear concept, guys? Now let's move on. So now let's know something about immunity. So immunization, this is basically the process of inducing immunity by stimulating immune system through antigens. Same concept here, guys. And now let's see immunity. It is the resistance of a host to a specific agent characterized by measurable and protective surface or humoral antibody and by cell-mediated immune response. So it is basically the ability of resisting the antigenic effect in the body. So this was the basic concept here. Now there are two types of immunity, and they are natural immunity and acquired immunity. Natural immunity, let's see, it is not produced by the immune response. This type of immunity is present at birth. From birth we have, so this is also called innate immunity and appears to be present in all members of a species. So clear concept here, guys. Now acquired immunity, it develops after birth as a result of exposure to an antigen, thereby activating the immune response. Acquired immunity can be either active or passive depending on whether the immune response takes place in a host or a donor. So it is basically acquired immunity or adaptive immunity which is given after the birth during the lifetime here. So let's move on. Now let's see the types of acquired immunity here. So acquired immunity, acquired after birth or during lifetime, and they are specific for a particular antigen. Means for a one antigen, one antibody is responsible to act on. So now let's see the types. So there are two types, that is active immunity and passive immunity here. So active immunity, antibodies are produced when antigens enter the body. For example, vaccine or immunity against certain disease after patients suffer from that disease. Very important concept, guys. Now passive immunity, in this ready-made antibodies are introduced into the body. For example, snake venom here, and this is the temporary effect, and it's and active immunity is the long-lasting effect. Now let's see why it is a temporary effect. Because in this suppose a snake venom here, whenever a person is snake bitten, then what happens, the ready-made antibody is given to the patient, means that a snake bitten person, and the antibodies acts on the venom and the person cures, and it is only for a certain period of time only. So whenever if a person in future is again snake bitten, then same antibody will not work, means again the ready-made antibody is to be injected. So that it is temporary, and this is the long-lasting phenomenon here, active immunity and passive immunity. So now let's move on. Now we need to understand some basic terms here, antigen and antibody. So antigen, we already know, antigen is the foreign material that elicits or increase antibody formation in the body. Very common concept, guys. Now antibody is the molecule synthesized by human body in response of antigen. It is also known as immunoglobulin, globulin or protein here, immunoprotein. This is also called immunoprotein. Okay, now let's move on. Now vaccine. What are vaccine? A vaccine is any preparation intended to produce immunity to a disease by stimulating the production of antibodies, and it includes suspension of killed or attenuated microorganisms or products or derivatives of microorganisms. Very clear concept, guys. Very easy definition here. Now let's move on. Now we need to understand how do vaccine work. Very important concept here, guys. So in that, what happens is, first of all, the pathogen, that may be virus or bacteria, is taken. That may be whole bacteria or virus taken, or some cellular fraction, means it's some protein, membranous protein, or genetic protein, or any body part is taken, and then it is processed, and after to that it is injected to a particular patient, which is affected by that pathogen. So against to that pathogen, our immune system creates antibodies and acts on it and fights on the fight it off with the pathogen, and the patient is cured. So this is the basic phenomenon. And in our immune system, there are many memory cells that stores that particular antibodies, and whenever in future, if again the same pathogen acts or attacks the body, then again the same antibody is used to cure the patient, so that we are not going to be sick in the future, and so we are called as immune now. So let's see the processes step by step. So here you are given a small amount of a harmless form of a disease, then your body makes antibodies to fight it off. Very good, same process as we have already mentioned. And then if you encounter the disease again, your body already has the antibodies, so you don't get sick, you are immune. So this is the common process we have already discussed. Very important concept, guys. Let's move on. Now we need to understand the types of vaccine here. What are the types of vaccine? There are 5 types commonly. Let's see one by one. So killed or inactivated vaccine. In this, what happens? The pathogen, that may be virus or bacteria, is totally killed or inactivated before injecting into the body. So this is killed or inactivated vaccines. And toxoid. Toxoid vaccine. In toxoid vaccine, what happens is the toxic property or the pathogenic property of a particular virus or bacteria is cut off, so that it won't infect to the patient when it is injected into the body. So this is the concept for toxoid vaccine here. And now attenuated or living vaccine. In this vaccine, what happens is the pathogen is not killed, it is only weakened, and the non-virulent property is created, means the virulent property is cut off, and then it is injected, and for that the immunity system responds, and antibody is made, and the patient cures. So this was the concept for attenuated living vaccines. Now antibody as vaccine. In this, what happens is the ready-made antibody are prepared, and they are injected into the patients, so that in a very short interval of time they can be cured. So this was the concept for antibody as vaccine. This can also be said as a similar to plasma therapy also. So now a structural or subunit vaccine. In these subunits or some part or cellular fractions are used for processing and formation of vaccine here. So now let's see one by one. Kill or inactivated vaccine, prepared by killing the pathogenic organism by heat or UV ray or alcohol or formalin. Yeah, for example, typhoid vaccine, cholera vaccine, hepatitis B vaccine, and influenza vaccine. Okay, now let's move on. Toxoid, prepared by destroying the toxic property of toxins produced by organism but retaining its antigenic properties. Same thing we have already discussed. That is, for example, tetanus and diphtheria here. Now let's move on. Other is attenuated living vaccines. Pathogen is made weakened and non-virulent, as I have already mentioned. For example, yellow fever, oral polio vaccine, Sabin, BCG, measles, mumps, rubella, MMR vaccine. Provide long-term vaccine. Yeah, of course. So now let's move on. Now antibody as vaccine. Serum containing antibodies against pathogen is used after a person or animal has been exposed to infection. So this is the concept, guys. In this, what happens? The antibody is collected or extracted from the plasma of a patient who has been recovered from the disease, and that antibody is used for the another patient which is infected from the same pathogen, and so that the antibody can act on the pathogen and the patient is cured. So this is the process of antibody as a vaccination here. Now a structural vaccine. In this, vaccine produced from extracted cellular fraction, that is subunit. For example, hepatitis B vaccines. Okay. Now let's move on. Now let's see about the classifications of vaccine. There are three classes, and they are first generation vaccine, second generation vaccine, and third generation vaccine here. First one is prepared by conventional technique. Second one is by recombinant DNA technology. Uh, and in this, DNA amplification is done. So this is one of the important process too. So, for example, your hepatitis B vaccine, herpes vaccine, pneumonia vaccine, and third generation vaccine, synthetic vaccine such as FMD vaccine. So these were the overall concept on classification of vaccine. Now let's move on. So now, once the vaccine is prepared, it has to pass some clinical trials, the phases of clinical trials, and there are four phases of clinical trials, and they are 1, 2, 3, and 4. In first, we have to evaluate the safety of vaccines, determine safe doses, identify side effects. In the second phase, test effectiveness, further evaluate safety. In the third phase, confirm effectiveness, monitor side effects compared to other treatment, collect information. And for the fourth, provide additional information about approval, including risk, benefits, and waste uses. And after to that, post marketing and distribution is done. So this was the overall concept for vaccination and immunization, and about their types, phases. So this was the overall concept. So guys, in our upcoming next video, we'll discuss on the COVID-19 epidemic. We will discuss and give some information about the vaccine, the diseases, and the coronavirus pandemic. So for today, this much only. There.